Okay, everyone, we can go ahead and get started. So thank you for joining us for this month's Science Cafe. I just wanted to start with a brief overview of the CTSC for those of you who are not familiar. So our goal is to improve the health of those that live in Northern Ohio and beyond by providing resources and services to our hospital and university partners, clinical and translational researchers and scientists, as well as community members. The CTSC is currently doing this by focusing on these 5 key areas and initiatives. So resources and services, workforce development, community engagement, health informatics, and clinical and translational funding opportunities. And I want to take a minute to introduce our presenter today. Dr. Sattar is a professor of biostatistics in the Department of Population and Quantitative Health Sciences at Case Western Reserve University School of Medicine. His research interests include longitudinal studies with complex problems, random effects survival models, joint modeling of outcomes, Bayesian semi-parametric regression for high-dimensional longitudinal data, and dynamic treatment regimens. As a biostatistician, he's interested in interventional and observational clinical studies. And over the past 12 years, Dr. Sattar has been devoted to applying biostatistical science to health sciences as a contributor, continual learner, and an educator. Today, Dr. Sattar will be talking about new innovative methods for clinical trials. So I will turn it over to you.
And thank you, Mackenzie, for this gracious introduction and for your invaluable support in organizing today's CTSC Science Cafe on clinical trials. I would also like to thankful and grateful to Dr. Mackenzie and the CTSC for the honor of this invitation. So I'll be talking about mostly clinical trials and adaptive trial. So let's look at the presentation outline. So here is the outline that mostly I'll be talking about the adaptive trial design, AD, and also be giving a specific example that recently I have worked on adaptive design for a sepsis trial. And we will be talking about a few methodological aspects of the adaptive trial design. This is a really exciting time to talk about clinical trial design because the new policy that NIH adopted on prioritizing the human-based research while reducing the animal use in research. That means there would be a need for clinical trial designs, innovation in the clinical trial designs, and etc. So again, it is a great time to talk about the clinical trial design and newer methods for conducting efficient clinical trials. So before getting into the adaptive trial design, I would like to introduce you the basic clinical trial designs. So what is clinical trial? A clinical trial prospectively assigns people to intervention to measure efficacy of, for example, of a drug on health outcomes. So there are basic study designs like randomized controlled clinical trials that could be parallel study design, also could be crossover. Other example could be cluster randomized trial. Most of the clinical trial designs are mostly RCTs, randomized controlled clinical trial, say in parallel manner. For example, we recently completed a study, Dr. Mackenzie was the PI, where we were looking at the effect of semaglutide in reducing fat in HIV patient population. So when we conducted the study, that was a RCT parallel study. We had two arms of that study. One was receiving the placebo, and the other one was receiving the semaglutide treatment for for abdominal fat matters in the HIV patient population. So as I said, most of the clinical trials are randomized in those I mentioned here, RCTs, parallel, crossover, cluster. What we have learned from these studies, basic clinical trial designs, they have some limitations. So the limitations or lessons that we learned from these RCTs, not only me, from the in general, I'm talking about from the literature that we found that when reality doesn't match the planning assumption. So when we conduct the clinical trials, before launching the trial, we estimate the sample size, decide the number of arms, and covariates that we would be adjusting for or matching that criteria that we would be doing. How about if the assumption doesn't meet in reality? Say, when we are estimating the sample size, there could be a situation we assume a distribution for an outcome variable with its mean and standard deviation. But if the standard deviation in reality, when we conduct the study, deviates from the actual or hypothesized value, what would happen? So misspecification can happen. It can impact the study integrity and the inferences. So these are the things in reality that we face in RCTs or basic clinical trial design. And the standard design could be slow and costly. One question per trial model means, for example, in our semaglutide study, GLP study, we looked at the effect of just one drug, one treatment on reducing the body fat and in HIV patient population. This is kind of standard scenario for most of the clinical trial design, basic design. Look for one treatment, one drug, one intervention, etc. How about if we want to study multiple drugs at a single time, single design framework, how we would do that? Also, another limitation we can say, average effects can hide who benefits. This is the scenario of heterogeneity in the population. So if a study population has some heterogeneity that I'll be talking more, well, we are currently working on a sepsis patient population. So in that sepsis population, I'll explain that more details later. The patients their inflammatory condition could be different depending on many things. The patient populations could differ biologically. Some other risk factors, say age, those are older age patient versus the younger, there could be the heterogeneity could be coming. So if you do not take care of this kind of heterogeneity and try to understand the average effect of a treatment, then we may miss the benefit. Who would be benefited for what drug? And another aspects of or limitation of the standard design is the inability to stop early. Say we, in a RCT standard, RCT randomized controlled clinical trial, if we are conducting the study for 48 weeks or 52 weeks, say 2 years, someone would be receiving the treatment, we cannot stop in the middle unless there is a serious concern from the DSMB report, etc. Generally, this randomized controlled clinical trial move forward until the end, and then we analyze and determine the efficacy of the treatment. So early stopping is not generally an option in the standard RCT or say cluster randomized trials. So as I mentioned, the distribution in the first bullet point was here. Misspecification of the assumption in the computation of the sample size, we assume certain mean and standard deviation. So for example, if you assume a study and have a endpoints and its distribution, say normal with mean and standard deviation, say 1, and we compute the power with a certain sample size, we say that with a given number of sample size, we would have a 80% or higher power with a standard deviation of 1. If the standard deviation deviate from the hypothesized value, say if it is 1.2, what would happen? The thing would happen is here. If you look at the power, would be compromised or it would be reduced. The study in reality or in actual situation, if after conducting the study, if you found that your study endpoint standard deviation is 1.2, then you'd be underpowered. And also on the other way, if standard deviation is smaller, then what would happen is your power of the inference is high, but we are using resources, time, patience, etc., or even say if a drug is truly efficacious one, we can stop early if we know that, and then give use that drug for everyday clinical use for patient treatment. So if you assign more people, longer duration for such a scenario where efficacy could be detected early, but we miss that opportunity. So these are the situations or scenario could happen if say, misspecify some of the parameters that we are assuming in designing the study. So this is an example. There are many aspects of the trials can be misspecified, and it can have an implications in the making an inference. So what we should do to address this type of issues, situations. So that is the thinking is coming, adaptive design. If we want to address those limitations, barriers, etc., that we just spoke or discussed, then adaptive design is an way to move forward. So basically, we are rethinking clinical trials. We are thinking for a better and faster evidence. So the question, when you hear adaptive trial, do you think it is a flexible and efficient? B, risky? So I'll let Mackenzie to conduct this poll.
So the poll should be up. And also, Dr. Sitar, could you put up your slides into presentation mode?
Oh, thank you.
Yes, thank you so much.
So answer to this, some of you say both, most of you said flexible and efficient, which is the thing. Basically, adaptive platform trial is a planned, pre-specified, flexible trial. And shall I stop the poll, Mackenzie?
Oh, yeah, I stopped sharing, so you should be able to exit out of it.
Okay. All right. So basically, again, adaptive platform trial design is a pre-planned, flexible trial design. So the word pre-planned means way before starting the trial, we have to specify everything in detail. So now I would like to present the details of key aspects of the adaptive platform design. So it is, again, prospectively planned modification to the design features. I am trying here to full screen. Okay, so what are the key aspects of adaptive trial design? Interim analysis is a key aspect of the adaptive trial design. We should and we would do a pre-specified interim analysis for understanding the study outcome and various aspects of the design and various aspects of the study. So it is prospective pre-specification, and as I mentioned, details should be outlined in protocol as well as the statistical analysis plan. Another key component of the adaptive trial design is the controlling type 1 error. We do not want to inflate false positive, that for sure. How do we control if we have a multiple interim analysis? There is a likelihood of if we do not correct it or adjust for it, there is a high chance for inflating the type 1 error, which we do not want to inflate anyway. And the estimation should be reliable and properly taken care of all the analysis that we will be doing in the interim analysis to final analysis. So why now? And again, it is flexibility. It is great and ethically also a sound one. We can stop early for efficacy or futility. So in terms of, say, efficacy, if we found that a drug is effective, then we can apply this drug in routine clinical uses. And it is also an efficient trial. In a sense, it would be minimizing the wastage of resources. So now I would like to share one example that we recently submitted. We are still waiting for the review outcome because we would have the study outcome, but because of the shutdown, we did not get the review outcome. So it is a biologically adaptive platform trial that we proposed for sepsis patient population. What we did in this study is, or proposed in this study, we will enroll a sepsis patient and we will do the real-time immunophenotyping, endotyping. So sepsis patients, some of the patients, so before getting into the details, generally in the sepsis patient, those are in the ICU, they receive antibiotics and resuscitation fluid. That's it, they got, irrespective of their, say, cytokine response. So in this study, we are proposing to do some endotyping, say endotype A or endotype B. We are saying basically some patients should be hyperinflammatory, some could be hypoinflammatory or immunosuppressive. So depending on their endotype, we will be giving depending on the endotype, then we will be assigning patients either in endotype this arm. Those are hyperinflammatory, either they'll be receiving, say, corticosteroid or placebo at the beginning. And then we will do the interim analysis. So after interim analysis, if we found that trial is positive, then we will be stopping immediately, and then we'll recommend for this methylprednisolone for routine uses. So same thing for those that are hypoinflammatory, we are planning, we will be studying GM-CSF and placebo, and also we'll do the interim analysis. So after interim analysis, again, if it is positive, then we will stop and recommend for this GM-CSF for routine uses. If it is negative, the trial analysis, interim analysis, we found that the say GM-CSF is not effective, then we will continue and look for other drugs. For example, for hyperinflammatory, we will study anakinra and compare with placebo. One aspect of the adaptive platform trial, generally single control group, but here we cannot use the single control group because those are in the hyperinflammatory. They are different from the hypoinflammatory group. That's why we propose two control arms. Say placebo would be here, also placebo would be for the endotype B. So next I'll, I guess, talk about more benefits of this adaptive platform trial. So statistical efficiency, more power or smaller N expected. And then ethical matters should be stop early for success or futility, and learning subgroup effects where the adaptive enrichment can be done in adaptive design, and practical aspect would be can be more acceptable to stakeholders. So these are advantages, potential advantages for the conducting an adaptive clinical trial design. And limitations, what are the limitations? Limitations could be the type 1 error. If we are not adjusting the type 1 error appropriately, then there is a high chance for false positive. So we have to be careful when we are conducting an adaptive trial design. We should apply all statistical aspects appropriately. And biased effect estimates, if the analysis was not done appropriately in a sophisticated manner, then naive analysis could lead to some biased effect estimates. And other may require larger maximum N depending on the conditional power that I'll be talking about it more later, and the longer planning term. So there are a couple of questions here for the audience, I guess. So answer to the first question should be B, response adaptive randomization. I'll be talking about more different types of adaptive randomization, like response adaptive randomization, covariate adaptive randomization, treatment adaptive randomization, etc. So this is a most important one in a sense that we have to be we have to pay attention to any response adaptive randomization that we do. It has a grave consequences on the trial if we make a mistake. And for stopping for futility, the answer for the second one is B, missing a delayed treatment effect. We can talk more on this later on. So as I mentioned, different types of adaptive randomization. We'll go a little bit details here. So design, the adaptive design methods based on non-comparative that I mentioned at the beginning on this slide is for calculating the blinded sample size. So sample size re-estimation can be done from the pooled data, irrespective of or breaking the treatment arms. So when we will say, if we are thinking about a continuous outcome variable, we have to always use standard deviation for estimating the various parameters. So the variance estimate need to be calculated. And if we calculate the variance estimates from the pool data, irrespective of treatment arms, that would be a non-comparative one. And it would be a blinded sample size re-estimation. And the other type is based on comparative data. These would lead different types of adaptation group sequential monitoring for efficacy or futility based on boundaries and alpha spending. And I'll be talking more boundaries and alpha spending in the next slide or a couple of slides down. And the sample size adaptation using interim effect. So sample size adaptation, again, conditional power rules, those are also technical aspects of the adaptive design. I'll be also presenting some aspects of the conditional power rules. So other adaptation is adaptive enrichment, biomarker-based one. The one we proposed adaptive, biologically adaptive platform trial is basically a type of biomarker-driven, biomarker-defined subgroup-based adaptive enrichment. And treatment arm selection, add, drop, these are also possible in a adaptive trial design framework. For in our case, you saw that if the trial is efficacious when we are conducting checking in the interim analysis, if we found that drug is effective, then we'll stop the trial on that arm and apply this drug to in clinical practice. So add drop arms is possible in adaptive trial design, which was not possible in the standard RCTs. And allocation adaptive, covariate adaptive, response adaptive, we talked about a little bit, but we have to be use of response adaptive randomization use would be very with a caution. It has a very significant issue if we don't use it. There are some mechanism to do it, how we will do it. Those are very technical probabilistic aspect called play with the winner model type of application. And then endpoint adaptation also in a adaptive trial design, we can switch, we can select endpoints or initially if we plan for a say single outcome variable, and we can move to another one, but those have to be listed or pre-specified before starting the trial and clearly stated. So briefly I mentioned here in this alpha spending and the boundaries. So the alpha spending is a technical aspect. So basically here FDA endorsed this type of alpha spending. What we are trying to do here is alpha, which is type 1 error. We want to preserve the type 1 error while we are conducting an interim analysis during the entire trial period. If we do multiple interim analysis, there is a chance if we have a multiple endpoints, multiple interim analysis, there is a high risk of inflating the type 1 error. How do we control it? This is a way to control it, means cumulative alpha span, means controlling the alpha while we are doing the interim analysis. So there are two methodology, O'Brien, OBF, abbreviated as O'Brien-Fleming or Pocock method. So one is stricter and the other one is, at the early, it would be very stricter and the other one is flatter. So depending on the information fraction, we can preserve the type 1 error while we are doing the interim analysis using the methodology of OBF or Pocock. And the other concept here is for more those conditional power. We can compute the conditional power generally recommended at the end of the study. Sometimes later of the study, say if we are conducting interim analysis here at observed Z, so observed interim Z, if it is at 3, then our conditional power would be higher for the if we are conducting at information fraction at 75%, for example. So this conditional power will be used in deciding whether trial would be moving forward or stopping it. There is an algorithm we will be talking a little bit later. So here, actually, it is in the next slide. So if we decide, or in a simple example, if we have a continuous outcome variable, we can compute the, if it is normally distributed, we can compute the Z statistics. So from the Z statistics, if it generally, if it is 1.96 above 96, we can say that it is significant one, and efficacy boundary, this is a boundary that we can decide 1.96 or whatever, maybe 2.5, or it would be decided before, in the protocol and the assay, SAP. And if Z computed Z in the interim analysis, at each interim analysis, if we found Z is greater than the efficacy boundary, then we can stop the trial. Otherwise, else we will compute the conditional probability. If the conditional probability is less than, say, 0.20, also this cut point would be decided ahead of time, pre-specified, generally 10 to 20%. If conditional power is less than a threshold, this threshold, then stop for futility. And if else it means if the conditional power is in promising range, say 40 to 60% or 0.4 to 0.6, then increase N to N max, that also decided before the trial or pre-specified. Otherwise, continue as planned. So this is kind of algorithm for making the decision whether, when and how to stop the trial based on the either boundary or both boundary and the conditional probability. So in a summary, adaptive feature, group sequential will solve the problem of stop early for success or futility. And we would need the interim outcomes and information fraction. For blinded sample size re-estimation, we can find the right size, and if variances or rates differ from the pooled variance or from the interim analysis, we can find the variance estimate and fix the issue of sample size. So this is a kind of whatever I have talked in previously, sample size adaptation, adaptive enrichment, the type of issue it would be solving from the interim, based on the interim data is in here. So next, I think I'll move to the software. What are the software available? So the free software R package is available for designing and analyzing group sequential adaptive design. The package name is called RPAC. And another popular one that it's not a free, nQuery sample size calculation can be used. nQuery can be used for sample size calculation in an adaptive trial design. And there are a bunch of other vendor provided softwares available for adaptive trial design. And now, few more questions here. So the answer to all of them are false. The first one is clear, it can go either way. And the second one, we have to adjust the confidence interval using the combination test. Bias adjusted or combination test should be applied here. And for the third one, so limit to a DMC or adaptation committee with firewalls. So for the, I think these are the final slides here, planning simulations and trial integrity. For adaptive trial design, we have to plan completely and prospectively. Number or timing of loops for interim analysis, exact adaptation algorithms, analysis methods and softwares, etc., should be clearly stated. And simulations are critical, especially if we are doing the Bayesian trial, then simulation is a way to conduct the study. And we have to evaluate the family-wise type 1 error, power, etc. For maintaining the integrity, document who can see, we have to document who can see comparative interim results, for example, DMC or adaptation committee, and when. These should be documented, well documented, and maintain firewall. So again, a few more questions here for Paul. So answer to the first question would be only with stringent boundaries and robust safety monitoring consider continued follow-up. Yes, it can be, but with a stricter boundary. And for the second question would be blinded sample size re-estimation on variance, accrual strategies, logistics, not comparative endpoints or randomization without rules. And the third question, what signal should trigger a pre-planned futility stop? And is fine if pre-specified. Explain pros and cons, predictive probability, what soil in Bayesian settings. For the fourth question, where would you place your first interim loop, early 20 to 30%, 40 to 60%, or late? It depends. Early for safety or futility, mid for sample size re-estimation, and late for efficacy or boundaries. So I think we are at the end of this Science Cafe presentation is about the funding opportunity. So adaptive design, as I mentioned, we just recently submitted, and there are more and more, I guess, would be coming. And I did not exhaustively search other calls, but here are the few information related to adaptive trial design. If you are developing methods, statistical methods, improving methods, PFA from the PCORI is available. And I think there are a few cycles in every year. And CLASER grants is available also. Adaptive method for the PCORI can be PCORI trial like CLASER one. This is a big one, can be applied. And we responded to this UG3, UH3, where Ken Remy was the PI, and I'm the PI for U24 portion. So this work was a very a good experience one. We are excited to hear the good outcome from the NIH. And there are a few references here related to this presentation, Adaptive Design for Clinical Trials of Drugs and Devices. And this is a FDA guideline. You can look at, this is a very good guideline. And there are other documents or literature available you can look at for the. So now I'll stop, and if you have questions, then we can talk and submit.
All right, so Abdus, first, thank you for that. Yeah, if you have questions, you could just say the question, or you could put it in the chat if you're a little shy. That would be fine. So Abdus, let me ask you one thing. It's actually little odd that the FDA in November 2019, as COVID is coming to our shore, they issue the adaptive design that ended up being very used in COVID. Maybe a coincidence, but it just eerie a little bit that it was the same time. Nonetheless, so all the COVID studies used adaptive design. How can you tell us, how do you decide when to do the analysis? Because as you mentioned, you don't want to do it too early.
Yeah.
Miss a long-term signal. So how do you do that? When do you decide, like in the sepsis study, when you do the initial analysis?
So for the sepsis study, we, since these are the ICU patient, so after giving the drug, either they will be cured or not cured, we do not, in the ICU, we do not have a long-term follow-up. So after, say, if someone is in hyperinflammatory, they would be assigned either placebo or a corticosteroid, and we will be following for 7 days. And this is the initial one we will be trying. After this one, we will conduct, I think we propose for 150 or something like this patients in 2 arms. And then we will do the interim analysis. If we find, if it is efficacious or not, depending on, we will be, again, another new sets of patients would be randomized for if it is moving forward with a continuation. But if it is effective, we will be stopping it. Generally, it depends on the fraction of information in the conditional power curve that I was presenting. We would like to, ideally, you would like to see it later in the study to maximize the power. But if the boundary decision is something also can play a role by itself, instead of the condition. So it can be by boundary decision. If it is an effective one, Z value can be, say, greater than 2, then we will decide whether a trial would be going forward or not. It depends on effect size of the interim analysis that we would be looking for, and also if the boundary value is not enough, then we would be combining boundary values as well as the conditional power to decide whether it's effective or not. So the timing generally later of the study instead of earlier, but it depends.
But you have to, I mean, I'm assuming it's predetermined, right?
Yes, it is
Time of
predetermined.
study Time, okay.
Predetermined, and depending on the expected, say, effect size that we would be hoping. And also the heterogeneity will play a role. If it is very heterogeneous patient population, then generally it will be towards the end instead of the beginning.
So I'm not gonna let you actually go without at least one AI question. So how do you see like AI, whether it's machine learning, models or any AI related analytics changing the way you do your job as a biostatistician?
Great question. I'm worried every day. All the white collar, this is the thing I say, all the white collar job may go away by AI because of the AI. So, but good thing is there are still many problems existing and need help. So in our study, the one sepsis trial, we are using AI, machine learning methodology, random forest for endotyping. The immunoassay that we would be conducting after getting the blood drawn, there will be a real-time immunoassay we would be doing. And after the immunoassay, we will do the endotyping. So how we will do the endotyping? We will apply machine learning methodology, random forest. So this is an application of AI.
Is NIH seeing it positively, you think, or you don't know? The review hasn't.
Yeah, they told us they will give us the review outcome before 1st of November, so that we can, if we have to resubmit, we can resubmit November 10th. But
Yeah. Wow. The government just opened up, so yeah. Maybe now you'll know. And let me ask you, since we are a CTSC, right? We're always here to encourage investigators, whether they want to submit a grant, use our services. How can somebody, to you in particular, but also like PQHS, like how do you see the CTSC interacting with your group? In other words, if we have somebody asking for statistical help, not just to analyze a study, but consultation for new research design. Like what among everything you talked about would be better if a clinician, for example, is explaining to you what the problem is, what to expect, what is their outcome, how do they get to you and your group to be able to talk to you before they submit a grant, to know the best research methods to use?
So I think through the CTSA, maybe one way to do it, listing my name, my group name somewhere in the CTSC, that I'm an expert on clinical trial design, especially the adaptive design. They can reach out to me or through your mechanism, CTSC consultation services. We have a shared resources here in the biostatistics shared resources. That could be another way to reach out. So we do not have, so I've been advocating for a data coordinating center. If we have one, then we know a data coordinating center should be supported to any application like this, but we are lacking it here. So the shared resources, PQHS leadership is saying that shared resources is a unit to go.
So by shared resources, and I'm not trying to put you on the spot, but if we have, let's say, among the attendees, some young investigator who wants to submit a grant and says, wait, maybe some of what Dr. Sattar talked about could be adapted in my grant. How do they go to somebody and they give them like a free consultation as long as it's expected they put them on the grant. That's what I'm trying to figure out, like, what are the
Yeah.
rules in the shared resources?
So for generally grant application, it is kind of free. The grant that I'm writing, helping, we do not charge, but with an expectation that if it is funded, there would be an effort allocation or budget. Budget would be specified, and then if it is funded, then we will be working together. But generally, that biostatistics shared resources, I think there is a I think commitment upfront, like cancer center biostatisticians, they are receiving their X percentages of effort for providing services to the cancer investigators. So I think similar mechanism in the past I heard from the CTSC members are receiving services and the biostatisticians are getting some percentages of their effort covered for these services, regular services that they are providing.
To do analysis, yes. So we do
Analysis.
have, yeah, just don't want to confuse actually the people listening. So we do have within the CTSC several statisticians to help with data analysis. We do a lot of Trinetics, a lot of, Cosmos coming up. So there are resources if they want to use large real world databases to have statisticians helping them. But I meant like outside even CTSC. So it is clear, obviously, if you help with a grant, you won't charge people, but hopefully they'll get the grant. So you get in it. If not, it is expected that somebody has to pay for that work, right?
Yeah, real analysis and work, someone need to find resources to compensate someone else's time, statistician's time. Yeah.
Got it. Okay.
Oh,
Any question, anyone? Oh, we have a quiet group today. All right, well, not bad. We have 7 minutes. So thank you, Abdus, again. And Mackenzie, I will let you close the meeting if you have anything to let people know, surveys and so on.
thank you.
Yes, I just have one slide to share. Okay, so these are just 2 QR codes. The one on the left is to get access to see all of the upcoming science cafes, so you can register specifically next month on December 11th at noon is the one on health informatics. And the QR code on the right is a way for you to sign up and become a member of the CTSC so that you can stay up to date with all of our upcoming events. And that is all I have. Thank you so much, Dr. Sitar. That was a great session.
Thank you so much for your all support that you provided. It was my pleasure.
Well, thank you, everyone. Have a great day.
Bye-bye.